Yesterday went okay. Or, sorry, Friday went okay. There was a little bit of a mix-up. I got the directions for her backwards, but they were right in classroom, so that's what you should have been doing. I'm sorry for all the cutting, all the intricate little shapes, but you won't have to do it anymore, assuming you did what you were supposed to do. So yeah, you've got replication down, right? You understand what happens there. Basically, the one enzyme splits the stuff open. The other enzymes run along in opposite directions, adding complementary bases in opposite directions, which means that the one that's going the same direction as helicase has that advantage of it's the leading strand and it just gets to go continuously, whereas the other one has to backfill and create those Okazaki fragments. Any questions about all that? Okay, so now we get into the beginning, at least, of what genes are actually for, what they do. What is that again? What does DNA actually code for? Protein. So that's it. That's what a gene does. But that turns out to be a lot. That turns out to be enough. Proteins are incredibly versatile, kind of workhorse molecules that, depending on what shape they are, can do a ridiculous number of different jobs. What are proteins made of again? Amino acids. You know amino acids better than almost any other molecule because you had to memorize how to draw one from scratch, remember? And so depending on which amino acids you string together, in what order, the finished protein will fold up in different ways. One of the powers that certain proteins have, there's an entire group of proteins that function specifically to help chemical reactions happen. Do you remember the names of those? Enzymes. Yeah, that's a lot. That's just one kind of protein, and yet that's what makes like every single chemical reaction that happens in your body, and that's you, man. I mean, that's like you are a big old cluster of chemical reactions happening at the right time, right? They're happening because of enzymes. There are millions of different human enzymes, all of them being made at exactly the right time to make exactly the reactions you need happen when they need to happen. Whether that's conducting nerve impulses, changing the structure of your cell membrane, breaking down your food. Every chemical reaction in your body is happening because an enzyme is catalyzing it. And enzymes are there because DNA codes for them. So that's just one kind of protein, and already we're taking care of all the chemical reactions of the body. Now, then there's what you're made of, which you know is cells. But what keeps all the cells, what holds them all together? What holds your shape? What makes sure you don't, slide apart into a pile of sludge? That would be proteins as well. Structural proteins are basically the glue that holds your tissues together. Let's do a little experiment real quick. You're going to need to take a real quick mask break to do this. Just pull your mask down. Wait, I'll show you. You're all going to pull your mask down, and then you're going to grab, okay, ready? Pull your mask down. Grab your lower lip and pull. Hard. Pull hard. Don't. Okay, good. So the reason you didn't just tear your lip off, the reason you could pull relatively hard on that thing It's real soft, right? Like it's kind of, lips are weird, kind of gross. Real squishy. But you can pull on that thing pretty hard and it's not going anywhere. It's not going to come off your face. What gives your skin that property of being remarkably soft yet very tough and stretchy is a structural protein called collagen. You've probably heard of it simply because beauty products companies love to talk about collagen and its partner, keratin, which is present in small quantities in your skin, but in larger quantities in your hair and fingernails. Beauty products love to put those names on their containers as it makes people think that it's scientific. Really, rubbing those things on the outside of your skin doesn't really do anything for you, but it is part of what your skin is made of. So just two jobs that proteins can do, and already we have all the chemical reactions and the glue that holds you together. There's way more. The point being that, yeah, DNA just codes for protein, but that turns out to be enough to pretty much run you. So that's proteins in a nutshell. The other thing we need to know is RNA. Right? RNA is the other kind of molecule we're going to bring in to get proteins built. And you're already aware, I think, of the first difference between DNA and RNA, because you're able to tell them apart in your paper shapes. What is the difference between DNA and RNA that you know already?
Like base instead of the deoxyribonucleic.
The thing in the middle of the nucleotide that both phosphate and the base are attached to, the 5-carbon sugar, is slightly different between the two of them, right? And the difference is literally just one extra oxygen atom in ribose as opposed to deoxyribose. That's probably worth writing down. So there's one difference. Have you noticed the other difference yet? The other chemical difference? There's one. The other three are the same. You still have cytosine, you still have guanine, and you still have adenine. But there's no thymine in RNA. Instead, there's what? To me, it sounds like something that you would take to help you go to the bathroom. What's it called? Close? Uracil. Yeah, doesn't that sound like something you'd see a really vague TV ad for? Are you having trouble going? Try Uracil. Yeah, so instead of thymine, it has uracil, which still pairs with adenine. It still does the complementary base pairing thing. It still forms the weak hydrogen bonds. But those two chemical differences then lead to a more physical difference. The chemical differences basically mean that RNA doesn't form as strong, it doesn't cling to a complementary strand quite as tightly as DNA does. It kind of peels off easy. And for that reason, RNA tends to be single-stranded. And it'll fold up into all kinds of different shapes, not just that double helix that we see with DNA. So it's a much more versatile molecule than DNA. It's almost like an in-between, like it's got information like DNA does, but it can do jobs like protein does. We're finding more and more jobs that RNA has. Turns out there's three sort of flavors of RNA. All three of them are going to be involved in the next processes we talk about. The first is messenger RNA. That's the one you already kind of know about, because we've talked about how DNA is used as sort of a pattern. We make a strand of RNA that's then going to carry the information out. That's messenger RNA. So the process we're about to dig into involves DNA being unzipped and copied onto a strand of messenger RNA. Messenger RNA leaving the nucleus and finding, what's the thing in the cell that makes proteins? Back from chapter 7. You're on a roll. Ribosomes, if you'll remember, are actually big old coiled up balls of RNA. And the kind of RNA that makes up a ribosome is called ribosomal RNA, or rRNA. Once the mRNA gets to the ribosome, which is made of rRNA, the ribosome kind of clamps down on it like a sandwich. And a third kind of RNA gets going as a shuttle molecule, going out and grabbing the right amino acids, bringing them to the ribosome and hooking them together in the right order. That's called tRNA, or transfer RNA. Questions so far? So this process of making proteins from DNA code that involves all three kinds of RNA is called gene expression. So gene expression, expressing a gene, means creating a protein using the DNA code. Ribosome. Yep. The big picture overview here. We simplify this as much as we can. We are looking at two phases, two parts of gene expression. The first part, the one you already knew about, where DNA, a section of DNA, remember we remember using the analogy of the blueprints and how you keep the blueprints locked in a room, but you have to let the workers in and out to copy parts of the blueprints, like maybe they take little pictures of parts of the blueprints and go build stuff that needs to be built. That phase is called transcription. That's where DNA is used to make messenger RNA, essentially copying part of the DNA code, one gene's worth, into messenger RNA that then is allowed to leave. Transcription, which if you know what that word means, to transcribe something makes a lot of sense. Transcribe, like if you were transcribing someone's speech, you're taking the information they're giving orally and you're writing it down in, like, written language. You're copying the information, but taking it from one form to another. That's exactly what's happening here. Copying the information from the DNA into another form, which is RNA. But it's still a code. It's a code for something else that's a different kind of molecule. So when you take that code that's in the mRNA and actually use the moving parts to build the protein that it codes for, that's called translation. You're translating the code into something functional. Okay, so from here, there's going to be a lot of details and quite a bit of drawing. We'll start it today. In the next, I'd say 2 days after this, it's going to be kind of drawing intensive and concept heavy, and you really want to have your A game. You want to have your thinking caps on or whatever. Okay, so we're going to dig into transcription now in detail. Remember, in replication, the copying of DNA to other DNA, we had two enzymes involved. We had the helicase, which splits, and then we had the DNA polymerase, which adds complementary bases. That was training wheels for this. The same two things have to happen, but you have one enzyme complex that does both jobs. So in some ways, this is easier because it only involves one enzyme. And that enzyme is called RNA polymerase. Not to be confused with DNA polymerase. But RNA polymerase has its own little kind of helicase part to it. So there's no separate helicase this time. RNA polymerase does the unzipping and does the adding of complementary RNA, except in this case, only on one strand, not the other. Basically, what it's doing is unzipping the DNA on its front end, adding RNA to the strand it's on in the middle, and rezipping the DNA on the back end. So there's drawing number one. We call the strand that's getting RNA added to it, we call that the coding strand of DNA, as opposed to the non-coding strand of DNA. And notice the letters, right? We said that RNA has a different base than DNA. Notice that what's matching up with A on the coding strand here is not T, it's U. Here's At any point here, if you have questions, please don't be afraid to ask them. I promise you're not the only one who has that question. Big difference here between replication, that we already drew, and transcription is that the DNA isn't getting changed at all. The DNA is just going to zip back up. And remember that mRNA isn't quite as sticky as DNA. What's going to happen here is that mRNA is just going to peel off and separate while the DNA zips back up. And we wind up with this separate single strand of messenger RNA we call the primary transcript.
Would deoxyribose be considered a sugar?
Yes.
Carbohydrate because it doesn't have a 1:1 ratio?
It's close enough, yeah. No, it's not always precise, as you saw when we did dehydration synthesis. That throws it off a little. So primary mRNA transcript, it's called that because it's not ready to leave the nucleus yet. We're going to need to modify that a bit before it can go, because all DNA contains junk, basically, leftover bases that shouldn't be there. And so you have to edit that primary transcript before it can leave. I think we'll leave that editing for tomorrow.